Hello, my name is Melina Nicou and my partner's name is DiPrima. Our research aimed to find a natural way to improve cognitive function within Alzheimer's disease patients. This neurological disease results in the progressive decline in memory and cognitive function. The disease that is among the most common causes of death within the elderly has 3 stages. Each stage has a different amount of symptoms. Each symptom can be measured on a scale called the Alzheimer's Disease Assessment Scale Cognitive. The first stage, mild Alzheimer's disease, progresses to include wandering, getting lost, trouble performing complex everyday tasks, and personality changes. The next stage encompasses hallucinations, difficulty in speaking, and completing tasks. In addition, this stage causes overall memory loss to worsen, which leads to a decrease in recognizing other people. The final stage, severe Alzheimer's disease, causes the patient's body to shut down, making them completely reliant on a caregiver. This shutdown is caused by the spread of plaque and tangles, which causes the brain tissue to lessen or shrink in size. The aforementioned plaque is made up of a deposition of amyloid beta proteins. This plaque, in addition to the neurofibrillary tangles, make up the causative agent of Alzheimer's disease. Amyloid beta proteins have shown to increase oxidative stress and inflammation within the brain. Nerve cells, called neurotransmitters, release specific chemicals. The transmitter associated with memory, which Alzheimer's patients lack, is called acetylcholine. Essential oils are known to have an effect on mood and cognitive function within adults. Mentha piperita, also known as peppermint, is known to increase the quality and speed and accuracy of attention and increase alertness. Seeing that amyloid beta proteins cause oxidative stress, we decided to use Rosmarinus officinalis, also known as rosemary, which is not only found to improve memory long term, but also to reduce oxidative stress within while having neuroprotective effects as well because of the carnosic acid it contains. The model organism we decided to use was Drosophila, fruit flies. Fruit flies are the model organism for neurocognitive disorders. In using Alzheimer's-induced fruit flies, we were able to measure the effects of essential oil on Alzheimer's patients, leading to our purpose. The purpose of our study was to find an effective method for improving cognitive function in Alzheimer's-induced Drosophila, so that it may be used as a possible model for humans. It was hypothesized that the essential oil Rosmarinus officinalis will have the greatest effect on the Alzheimer's-induced fruit flies' cognitive function in comparison to the essential oil Mentha piperita, because it has various neuroprotective and antioxidative effects against amyloid beta toxicity. Cognitive function will be assessed by the ability to fly up the chamber in the ring assay. The statistical hypothesis include the null, which would mean there is no statistical difference between the height the wild type and Alzheimer's-induced fruit flies are able to fly up in the ring assay chamber with the addition of essential oils. The alternative would mean there is a statistical difference between the height of the wild type and Alzheimer's-induced fruit flies being able to fly up the ring assay chamber with the addition of essential oils. Our experimental setup included placing 10 wild-type flies into 3 separate tubes. We decided to label these 1WT, 2WT, and so on in order to avoid confusion. We then placed 10 Alzheimer's flies into 3 separate tubes, which were also labeled. In tubes 1WT and 1AD, we added 1 milliliter of Rosmarinus officinalis on a cotton swab for 1 hour. In tubes 2WT and 2AD, we added 1 milliliter of Mentha piperita on a cotton swab for 1 hour. We then left the tubes 3WT and 3AD untouched as a control. In setting up the ring assay, we had to assemble vials with 3 flies from each wild type group into the ring apparatus. We allowed the flies to acclimate to the environment undisturbed for 5 minutes. During this time, we were able to set up a camera about 1 milliliter in front of the apparatus. In doing this, we were able to focus and zoom the camera onto the apparatus once we started recording. We then carefully took hold of the ring apparatus and sharply tapped the apparatus down on the surface of the bench 3 times, ensuring that the tap is hard enough to knock down all the flies to the bottom of the vials. After all the flies have reached the top of their vials, we stop recording. After completing 10 trials, we observe the video and document each fly's height climbed at 3 seconds after the 3rd tap. We then assembled vials with 3 flies from each remaining group into the ring apparatus and repeat the above steps. This is what the ring apparatus looked like while we were recording.
Hi, my name is DiPrima. I apologize that my camera is not working and you cannot see me. I will now discuss the results for this study. In Table 1, the height climbed in the ring assay, the mean height, standard deviation, standard error, and range values are recorded. In Figure 6, the average height climbed for the various groups of flies, as well as standard error bars are shown. In Table 2, the unpaired t-test and single-factor ANOVA p-values are recorded for wild-type control flies and untreated Alzheimer's disease flies, wild-type control flies and Menthopiperita treated wild-type flies, wild-type control flies and Rosmarinus officinalis treated wild-type flies, untreated Alzheimer's disease flies and Menthopiperita treated Alzheimer's disease flies, and finally, untreated Alzheimer's disease flies and Rosmarinus officinalis treated Alzheimer's disease flies. The comparison of untreated wild-type and Alzheimer's disease flies shows that there is a pre-existing difference in cognitive function, allowing for valid tests of the effects of the essential oils. For wild-type flies, only those treated with Menthopiperita showed an improvement in their cognitive function. This was supported by non-overlapping error bars and a p-value of less than 0.05. This shows that there is a statistical difference in the height climbed between flies exposed to and not exposed to Menthopiperita. For Alzheimer's-induced flies, both essential oils showed to improve the cognitive function of the flies. This was also supported by non-overlapping error bars and a p-value of less than 0.05. This shows that there is a statistical difference between the height of Alzheimer's-induced flies that were exposed to and not exposed to essential oils. However, our hypothesis was not supported, as we believe that Rosmarinus officinalis would have a greater effect on improving cognitive function. However, it did not. While carrying out this experiment, there were some limitations that we encountered. The amount of CO2 gas used to anesthetize the flies was not kept constant. This could cause a difference in the time for the flies to regain consciousness, which could later affect their ability to climb in the ring assay. We were also unable to determine if the essential oils were absorbed by the flies, which means they could vary in their exposure. There's also a lack of knowledge on the effects of the concentration and amount of essential oil, as well as the duration of exposure that is suitable for the flies. In the future, many variations can be applied to improve this study. For example, female flies can be tested in addition to males. The flies can be exposed to the oils for different amounts of times. Other oils, such as ylang-ylang oil, can be used. Diets of flies can be altered, and medications can also be used and tested using the larval crawling assay. Thank you.